The next presenter is Kareem Daari, and he's from the University for Development Studies, Tamale.
I have an interesting story to share with you. That is part of my PhD journey in the University of Aberdeen in public health. The background to this story is that research in Ghana in 2009 indicate that, by the International Livestock Research Institute, indicate that most of the ready-to-consume fermented milk dairy products in Ghana fail to meet the quality standards of fecal coliforms, indicating that most of these products were not processed under hygienic conditions. And the poor hygienic conditions under which milk is extracted in Ghana, processed and marketed, is a major constraint towards the consumption of safe fermented milk products in general. Therefore, in September 2012, with funding from the Department for International Development, UK, the International Livestock Research Institute gave birth stocking to 3 institutions in Ghana to do a training for milk producers and processors on personal hygiene and good manufacturing practices. The institutions were Kwame Nkrumah University of Science and Technology, MOFA, Women in Agricultural Development Division, and the Council for Scientific and Industrial Research, Animal Research Institute. This training was to reduce the public health risk of coliforms and other pathogenic bacteria in fermented milk products in general. So my study, therefore, was to assess the presence of coliforms and other pathogenic bacteria obtained from trained and untrained milk processors in the northern part of Ghana. What methods did we use? We collected samples, 10 samples from trained and 10 samples from untrained milk processors, Nunu samples. And those selected for the collection of the milk samples were also interviewed using a semi-structured questionnaire to assess how they routinely process this milk, including the batch we're collecting the samples from. The samples were collected and transported to the Animal Research Institute by air in sterile stomacher bags packed under ice in ice chest. And the samples were stored at -20 degrees Celsius in deep freezers at the Animal Research Institute in Accra here until further processing. So at the Animal Research Institute, what we did was to assess the coliforms in the samples collected. And subsequently, we needed to transport the samples to the United Kingdom for further analysis using molecular biological methods. However, the UK government said Ghana was a high-risk country in terms of importing these animal products into UK. So what we did was, we were given license to come and extract DNA samples from these samples before we could do further analysis in the UK. So I came back to Accra, extracted the sample, sent to the UK by courier, and we did use molecular biological methods at the Red Institute of Nutrition and Health, University of Aberdeen. And part of the samples, 10 out of the 40 samples, were sent to Teargast Institute in Cork, Republic of Ireland. What came out from the culture methods that we carried out at the Animal Research Institute in Accra here was that 50% of the samples collected from untrained milk processors contained coliforms, including E. coli. And 75% of the samples collected from trained milk processors contained coliforms, including E. coli. So there was no significant difference between the proportions obtained from the trained and untrained milk processors. And at the Wright Institute of Nutrition and Health in Aberdeen, we use molecular biological methods, denaturing gel gradient electrophoresis to assess these samples for other coliforms and potential pathogens. And 10 out of the 40 samples we extracted DNA from was sent to Teargas Institute, Northern Ireland, Republic of Ireland, sorry, for next generation sequence analysis to determine the actual bacterial content of these samples. The results we found from the molecular biological methods used at Wright Institute and Teargas Institute in Cork, Republic of Ireland, was that the most prevalent bacteria in all the samples were streptococci. And there was also high abundance of Shigella or E. coli or Enterobacter, often exceeding 10% of the total bacterial content of the samples. And what are the implications of these findings? The implications of these findings are that looking at the high coliform counts present in these samples, it means that a lot of the processing and assessment of the milk samples were done under poor hygienic conditions. And looking at this story in the context of assessing the feasibility of using fermented milk to improve complementary feeding practices in northern Ghana, the milk samples collected, processed as at now, could have their consequences if it is used as complementary food, because these samples contain high coliform counts and it could increase the risk of the children getting diarrhea and other infectious diseases, which we all know are risk factors to child undernutrition in low and middle income countries. Therefore, there is the need for us to look at more effective training methods that could improve the safety of Nunu to promote it as a complementary food. Alternatively, we could look at other alternate processing methods that could be used to improve the safety of Nunu, because Nunu is a traditional fermented milk product that is processed under ambient temperature conditions. Milk is allowed to ferment in containers, calabashes or plastic containers at ambient temperatures without the use of starter cultures. And it was also found out that most of the women use and sterilize containers to do the fermentation of the milk. And there was no cooling of this milk along the processing chain. So all these handling and processing techniques pose public health risks to the consumers of these products. Therefore, we need to make extra efforts to improve the safety of these products, to promote it as complementary food, to improve the nutritional status of children under 5 in the northern part of Ghana. I wish to acknowledge all those who helped me from the various laboratories to assess these results. Thank you very much.